This thesis presents a creation-centered explanation for human diversity by integrating biblical genealogy with genetic science. It argues that the origin of humankind lies in the Mesopotamian region, near the biblical Garden of Eden, rather than through evolutionary processes. African populations retain the richest genetic diversity not as a product of mutation, but as a preservation of the original human design. Through an analysis of ancient lineages, particularly Ham's descendants and the Egyptian pharaonic bloodline of Mizraim and Ramses III, this framework offers a compelling, faith-aligned alternative to evolutionary theory. The genealogy in Genesis 10 records that Noah had 3 sons, Shem, Ham, and Japheth, whose descendants repopulated the earth after the flood. Ham had 4 sons, Cush, Mizraim, Put, and Canaan. Although Ham's lineage begins with 4 sons, the record of their descendants is extensive and geographically influential. Mizraim, traditionally understood as the father of the ancient Egyptians, is of particular interest. Modern genetic testing platforms, such as 23andMe, have traced paternal lineages back to Ramses III, suggesting that contemporary individuals may be directly descended from ancient pharaohs. Ham's other sons also played major roles in ancient civilization. Cush is often linked with regions of Nubia and East Africa, Put with areas west of Egypt, and Canaan with the Levant. The curse placed on Canaan did not apply to all of Ham's descendants, but it did carry generational consequences that affected the geopolitical landscape of regions historically associated with the Canaanites. In a modern context, the ongoing struggles in the lands historically linked to Canaan reflect, to some extent, the social and historical consequences of that judgment, even if the spiritual implications no longer apply. From a genetic standpoint, African populations possess the greatest diversity in the human genome. This is not because of evolutionary processes that suggest man emerged from primates, but rather because these populations remained geographically closer to the region of origin and preserve the original human blueprint. Adam and Eve, as the first humans, would have carried the full range of genetic possibilities. As their descendants spread out, especially after the dispersion at the Tower of Babel, they became more isolated, leading to genetic bottlenecks and a reduction in diversity. This explains why African populations retain the capacity to produce a broader range of human traits, including features commonly associated with other ethnic groups. This understanding offers a harmonized perspective between science and scripture. The genetic richness of African peoples aligns with the concept of proximity to the source, not progress through mutation. It affirms that humans are one family, not evolved from animals, but designed by a creator. The variations in appearance and culture are outcomes of environmental adaptation and genetic sorting within a divinely orchestrated plan. This thesis presents a biblically grounded, scientifically aware model of human origin. By recognizing Mizraim as the progenitor of the Egyptian civilization and potentially linking his descendants to present-day individuals through genetic analysis of Ramses III, we affirm the enduring accuracy of biblical genealogy. Furthermore, the observed genetic diversity in African populations supports the belief that they retained the original, rich human genome. In contrast to the claims of evolution, this model respects divine creation, preserves human dignity, and emphasizes the shared ancestry of all peoples under one creator.